So, so Chimay already did a little bit of introduction about me, but yeah, I had this slide. So yeah, let me skip the first two lines. Yeah, so first thing, it's like I founded this Arabic Neuroscience initiative. So yeah, more about this later. And yeah, my main current interest, as you probably can guess, is computational neuroscience in mental health. And a couple of fun facts about me. Yeah, I visited Fukushima, neural clear exclusion zone. And not fun fact is I live in Canada currently, which is too cold. Not now, but in general. Okay, so today we're going to be first going into a couple of stages. So first we're going to be talking about like actually what do we know about human vision, and then about computer vision, and then how similar are those like two domains, and then how can we actually use under like artificial intelligence to understand how the brain processing is happening. And then what can the synergy between human and computer vision help us to understand mental disorders? And mainly it's going to be lessons from schizophrenia. So let's start with human vision. So basically our human vision system, of course, we start with the eye. And basically what happens is that there's the light information. So light with different colors is captured by our eyes. And basically our like the receptor cells, there we have two types of them. You can see here like the cone cells and the rod cells. The rod cells are mainly responsible for things that are like just black and white vision. So they're not really into color sensation, while the cone cells are the ones that actually you can actually watch color from them. And then they send input to bipolar, in turn goes to the ganglion cells. And then all of that travels using the optic nerve through the optic chiasm and the lateral geniculate nucleus, and then to the primary visual cortex. Don't worry about all of those names. We're not going to be, it doesn't really matter what their names are. But the idea that we want to focus on is like, if you look at the thing, like the graph on the left, you'll find that the cones are really centered. So the zero here means like the zero angle of where you're focusing, basically. And you can see that all of the color information or color sensing cells are really concentrated at the middle of your focus, while the rods are not, as they're more distributed towards the peripheral vision. So things that are, you're not really focusing on, but like the surrounding vision around you. And that's one of the reasons why we actually focus a lot. We use actually like our focus is really important to us. Like peripheral vision, we don't really see what is happening. You kind of see like a blurred kind of vision. And we'll see even more about that in a little bit. Yeah, so as I said, we have like those two types of cells, one called rods and one called cones. And this is like an electronic microscope image. And then we can see here also like what colors are the different, there's like three different types of cones. One is seeing the blue, one seeing greenish, yellowish, and one that is like more towards like the reddish side. And then those cells, what they do is that, as we said, like they move to the bipolar and then the retinal ganglion cell. So the difference, the retinal ganglion cells, they're different from the rest of the other cells is that they actually emit spikes. Spikes, the good thing about those like spikes, basically if your cell is receiving a lot of input from the cells before them, and or basically which is the electric signal from the light, and if they pass a certain threshold of activation, then it emits a spike, which is basically, think of it as you're switching the light on and off really quickly. The good thing about those spikes is that they can travel for a really long distance, which is what they're going to be doing through the optic nerve. So think of like the distance between the end of your eye and like the LGN part in the middle, and also next is the primary visual cortex. And they have very interesting like different types, and they have very different interesting characteristics. So you'll find different types of them. One of them is on and off on center. So basically, like if you shine a light in the middle, then you're going to get like a really strong response. But if you shine a light at the off area of like basically each cell is responsible for different parts of your vision field. And if you light at the off part, then you're going to actually decrease the spiking the spiking rate. While if you shine more light on the on part, then you're going to get like a lot more spikes. And also if you shine light on the outside part, which is supposed to be off. So basically the idea is that the on center means that if you have more light, then it's going to be emitted more, but the off surround, you need to have less light to actually have something. And the off center is basically the opposite. It focuses on like the dark center and the lighted background. So those are the two types. And this is like the first level of processing. It's just processing this kind of like a dot that is surrounded by like a black dot surrounded by a white light, or a bright spot surrounded by a dark background. And then, then there's like as we said, it sends the input to this area called the primary visual cortex. So this primary visual cortex is like the first part in what is known as the cortex of the brain. So the cortex is where a lot of what makes us human basically is located. We have like all of this cortical surface, which is like one of the largest relative to our body sizes. And this is where a lot of like this neural processing that is happening that's very different from the rest of the animals. But it's also, you can find this cortex it exists in a lot of different animals as well. And one of them is the cat. So there is this really interesting experiment that happened in the 50s by Hubel and Wiesel. And what they did is that they put electrodes to measure the electric activity in this area that's called V1, which exists also in cats, exists in humans, it exists in monkeys, you find it in many different mammals. And first what they did is that they showed the cat a screen, and they shined dots of light on the screen. And then nothing happened. And they were really disappointed. There's like nothing, they're getting nothing from like any electrodes. And then what they noticed is that when they were removing the slides that showed the different dots, they found that actually when the motion of the slides was actually starting to give them a response. And what they noticed is that the edge of the slide as it was moving, that was actually the thing that was eliciting a response. And then what they discovered is that they have different, when they move a certain edge-like shape through the screen with different orientation, like each orientation, you have a different types of cell that is actually getting activated, that is showing spikes. And basically their way of measuring spikes, they had actually an audio. So it's kind of like if a cell is spiking, you hear like this sound, and more spikes means like this gets more intense. Yeah, so then emerged this idea of the orientation selectivity. So some cells are selective to a certain orientation of the edges. And this is what we would say would be like the second level of the processing, but inside of the cortex, it is considered to be the first level of processing inside of the human visual cortex. And this area is known as primary visual cortex, or V1. Yeah, and the interesting part is that when you look at how the V1 actually represents the stimuli, they're actually very topologically accurate. So what is actually projected on this part of the brain is like, you can map it, like it's a little bit distorted, but you can still map it to what you can see in the actual image. So for example, this image was shown, and this is a flattened part of the V1. And you can see like all of those stimuli are still mapped, but although they are distorted because they look a little bit different. But in the end, they still like, you know, parts that are beside each other in the actual image are still beside each other in this visual cortex. So it's kind of like you're creating a visual map inside of your brain. Yeah, and as we said, the part that is in like your focus, or like the center of your focus, is actually more important. That we saw like this really high concentration of cones. So this difference is also mapped inside of the brain. So you will see that there's a large part of this V1 that is actually concentrating on the center of your focus. So if you're looking at the image on the left at the exact center, then the map is going, the cortical map is going to be looking like this. What's like in the center is like being processed by a larger number of neurons than whatever is in the periphery of your vision. So that's the primary visual cortex. And then we noticed that the visual cortex in the first part was only looking at things that are, like the orientation of edges and lines and things like contrast. And this is a very low level of vision. But then if you are actually able to like measure edges with different orientations, then you can actually make shapes. And that's what the V1 is sending an image into what is known as V2, which is also the second level of cortical processing of images. And then that sends to other visual areas progressively. So there is actually more than what you see in this image, but this is what is known here, is what's shown here is known as the ventral stream or the what stream. Basically, it is responsible for knowing what is in the image, what is the content of the image, understanding the identity of what you're seeing. And what happens is that you are progressively adding up information. You're doing like in a hierarchical fashion. You first process very basic information, such as like the edges, and then from the edges and lines, you make shapes, and then from those shapes, you make objects, and then from that, you make things that you actually know, such as faces or objects or scenes or even like body parts, whatever. Depends on, of course, what is important to us. And that is known as the inferior temporal cortex. That's the last part where you can actually find those areas. Yeah, so you'll find that when we look at something that is even like more fine-grained, we have what is known as the lower visual cortex, which includes like this V1 and then also V2 and V3. And then they send information to this V4. And then the IT is composed of different specific regions. So one of them is LOC. LOC is known to be recognizing objects. For example, FFA is known to be recognizing faces, and PPA is known to be recognizing scenes. Yeah, and you will find actually there is like different, there are different studies that, they show faces and then they show scrambled faces, and then they find like this part of the brain that they call the FFA. This is the part that is actually like lighting up really strongly. And then they show the scenes, and then I find this other part, PPA, is really strongly activated. So that's how we as humans, or as the human brain, what it does is that it does this hierarchical processing of the image. So how does the neural network do it? So or how does computer vision do it? In the past, actually, computer vision was like a very difficult thing. So what people used to do is that they would see the image and then it would look like, yeah, how can I actually turn this into something that the computer understands? So for the computer, images are just a bunch of numbers. So like basically each dot, like the image is divided into small squares. Each square is called a pixel. And then each pixel has a certain value. And the mixing, for example, also it has actually each pixel has more than one value, one for the red, one for the green, and one for the blue. And then the mixture of those colors is what creates color images on the computer. So the computer, what it sees is that an RGB value for different pixels in the image. And yeah, so processing that for the computer is really difficult. What people used to do in the past is that people who are interested in image processing would be like, yeah, this thing looks like I can divide, I have to apply this complicated mathematics to be able to divide it into something called features. And then those features will start putting it into a machine learning model. And then this machine learning model is like, oh yeah, I found this feature here and feature here, and then this feature here. And then they combine as okay, that's probably a car, or that's probably not a car. But then recently what happened is that in 2006, the model on the right was developed, where it was one of the most difficult things, how do you actually recognize written character images in a computer? And then there was this deep learning model that was developed by Jan LeCun at the time, and that's called Linet. And that was like a revolution in this idea of making actually a model that has many layers of classification or of processing of a certain image on the computer in order to be able to classify it into different identify or understand the characteristics or the content of the image. So in this case, the deep learning doesn't really need this expert person to decide what is important in the image as of like features, but what it does is that it picks those features by itself. And it was basically like, this idea was not new. It was actually really old, but it's just that computers were not as good as they are now, and there was not as much data to train those networks. Yeah, and then in 2012, so every year they had this thing called the ImageNet competition, where basically people will, like, there's this really huge image data set. It's contained like 1.3 million training images and like 50,000 test images. And what people would do is that it has 1,000 categories, and the people will submit their models and test those models on how to classify those images into one of the 1,000 categories. And you can see that the area, like in 2011, here on the right, in 2012, you can see all of the models here are having error rate around 0.3, even more, up to 0.5. And the thing that is between them, all of those are not deep learning model. And then come 2012, there was this model that just basically destroyed all of the competition called AlexNet. And it was like over 10% lower than the best model like in all of the years before. And since then, everyone just jumped to the bandwagon. It's like, okay, that deep learning seems to be successful. Let's make even better models. And that's even 2016, they just stopped making this competition because it just becomes so easy. And what this AlexNet did is that it created like 8 layers of processing of the image. So you insert an image, and then it starts, it applies a certain process called convolution on the whole image. And then it starts, you can see, if you look at the outputs of each of those layers, then you can see that what is being recognized is something that is a little bit more complex than the layer before it. So if you look here really closely at this conv1, you'll see like things that are like black turning into white. So think of like white as like a high level of light and black as a low level of light. So they're actually recognizing something that looks as edges. And then if you go deeper and deeper, you find that it's starting to find object parts, like in conv5. And then in the last layer, it actually recognizes like the whole object or the category inside of that. So how does it do that actually? So if you look at the output of like the first layer, conv1, so this guy is part of a video where they were looking at what is the output of different layers. And yeah, conv1, basically, this is the output of the first layer of the neural network. And the input is this image of the guy in the camera. And if we look at those two boxed outputs, so there's multiple outputs that are coming from like the first layer, but then we can look at each one of them is applying a different processing technique. But if we look at those two, then we'll find that one of them is picking like the right edge of his face, and one is picking more like the left edge of his face. And if we test this further, what he did here is that he started holding a paper. And then that paper goes from light, like white, to darker. And you can see that the one is circled in blue is the one that is actually picking up that edge, while the other one is not picking it up at all. But on the other hand, if we have the papers in the opposite sides going from dark to light, we find that the red box is the one that is actually picking up. So one of them is picking the rising edge, and one of them is picking the falling edge. So we can see that this layer is doing like really basic stuff, like just basic recognition of something that is very simple. But then if we go to something that has conv5, we can see that this filter that is recognizing his face. So like when the area around his face is what is labeled in this output. And then we'll find different filters. Some of them might be still recognizing face, but because it's recognizing something else and other things. But yeah, mainly you'll find like this kind of earlier layer is picking up something really simple, such as an edge, while a deeper layer is picking something that is much more complex. And it goes through all of those layers in the different like conv1, conv2, conv3. This goes really progressively. So what we will notice here is that there are two systems that are doing something really similar. They're basically processing an image in a very progressive fashion, trying to pick up something like edges in the beginning, like as we saw here in the lower visual cortex, and is the same thing for this deep neural networks. But at the same time, it starts as we go deeper inside of the visual cortex or the deeper neural network, then you're going to find that the processing is actually getting a little bit more complex, and like you're going to be picking something that is actually meaningful to you. Yeah, so the question, let's get back a little bit. I noticed that you were actually talking about that in the last talk, is the idea of like, how do we actually measure those things in the brain? Yeah, so the method that we're going to be using like from now on in the rest of the presentation is called fMRI, or functional magnetic resonance imaging. And what it does is that it tries to measure the activation of the brain by using magnetic fields. So what it does is that there's like, for each molecule in like whatever, like all of the molecules, there is a certain resonance frequency that if you apply a magnetic field with a certain frequency, then you're going to have like a very strong vibration. And yeah, then you can read out how much of that. And basically you decide what you want to read based on what frequency you're applying. So there's MRI, which measures the brain, like the anatomy of the brain, which basically what it does is that it's either looking at the water or it's looking at the fat molecules of the brain. While in fMRI, it's trying to looking at hemoglobin molecules, which is oxygenated blood, because an activated neuron needs more oxygen. So it will be receiving more oxygenated blood. And that's basically the proxy that fMRI is using to measure the brain activity. And yeah, for each part of the scan, we get like something like a time series of that. Like what you do is like you're scanning the whole brain for like, a few seconds, like 2 seconds or something, and then you do it again, you do it again, you do it again, and then you have a time series. Of course, it's a very slow process because you have to scan the whole brain, but you get decent information from that. Yeah, so then if we know actually, if we can measure how the brain is like how much the brain is activated, then what we can do is to actually measure, like we can know what you're looking at just by measuring your brain activity. And that's what was done in this study. So yeah, so this study is actually from it's from 2008, but it's the same lab that I did my PhD in, but long before I joined. And the idea is that they're trying to reconstruct what you're seeing just by looking at your brain data. And it was quite successful for the abilities at the time. So you can see here in the video, like you present this image of a square and then you get a reconstruction of it. And the same thing, you start to present different images and you can see the reconstruction it's not as good for every type of images, but it is relatively good. And it was something out of this world from the time. Okay, now we have a brain image, or like the brain data that we know processes the image in a certain way. And we have the computer vision model that we know is processing also in a certain way that is quite similar, at least from theoretically. But how can we actually test that? So this is one of the idea, also like same lab, but also before I joined. So what they did at the time was they actually have one of those neural networks, which is incidentally, it is AlexNet, which is the one that was in 2012 that, like started this whole revolution. And what they do is that they show the person inside of the fMRI scanner, like those images. They show like many of those images, and then you have the brain data. And then from the brain data, you try to measure, like create a measurement to predict the output of different layers of the neural network. So you remember like this conv1, the output of it, the conv2, conv3, conv4, etc. until the last layer. So here these are CNN1, 2, 3, up to 8. So CNN1 is like the one that is closest to the image, and 8 is the deepest one. And then they try to predict that from the brain data. So you're basically projecting the brain data and the neural network data on the same space. So then you can actually compare it. And then the interesting part, then you can actually measure how similar they are. And what they found is that this V1, V2, V3, do you remember the ones that are most shallow, or like the ones that are measuring something very simple? They're also like highly correlated with the shallower layers of the neural network. But the FFA, PPA, LOC, like the deeper parts of the human visual cortex, they are correlated with the deeper parts of the neural network. So from this, we can see actually indeed that the neural networks are doing something really similar to the brain. And this, like, from using the data. So it is like we can actually use those deep neural networks as a proxy for how the human brain is processing an image, right? And that actually led to, an improved reconstruction. So you remember the first study where we were showing the reconstruction of the image. You can also get that even better by applying this whole idea of translating the fMRI data into the neural network space, and then you try to reconstruct the input image from this data. And yeah, you can see the reconstruction from like this is like a very simple shape. So it's natural that the V1 and V2 are the ones that are having the most faithful representation of those images as compared to like V4. HVC is basically high visual cortex. So it's all of like the LOC, FFA, PPA areas. So then the question is, if those two systems are really similar to each other, then the question is like, is our vision that simple? And the answer is no, it's not that simple. It's like everything in the brain is really highly connected, that it's really hard to actually like correlate the connections together. So there's like the feed forward connection, or the one that is going to like from image to understanding or categorization, that seems to be homologous. But because of like all of these connections, is actually the human brain is much more sophisticated, and it has a lot of more functionality. So the neural network are like represented by this called pure bottom up. Each layer is receiving the output of the layer that is before it. But the brain itself because it has also like this recurrent connection, the layer itself, the output of it is fed back to the same layer, and the output from the top and it's also returned back, etc. So you have like all of those feedback connections plus the existing already feed forward image. And what that leads to is that our vision is actually quite robust. So when we are presented with something that is partially visible or cluttered information, our brain is able to use this feedback drive to complete this information from the stored representations. So because we know, for example, how a dog or a cat look like, so we can actually fill in the information in the middle that are missing from the image or not really visible. While, how does the computer actually do that? So here, this word cloud is like basically this, the bigger the size of the word means like the higher the probability of the classification of that network. And you remember we said there's like 1,000 image choices or possibilities. And you can see here like tabby or tiger cat, Egyptian cat are like the most, like the biggest classification probabilities. So the neural network is doing really fine. But let's see what happens if we actually start blurring the image. And you'll see that quite quickly, it actually, let's see, go again. Quite quickly, it starts to fail. It starts to recognize all of those weird things, even though we can actually still see a cat at this 6% blur level. And then if you go further, you see it starts to recognize a milk cat. I think most of us can still see the cat. And yeah, you can see that this neural network is not very robust. Once we start to perturb the image a little bit that is away from what it is used to, then it starts to just fail completely. So then this is when I got in to study. I wanted to see, so how is the brain different from those neural networks? So in the MRI scanner, we presented those blurred images to the person. And then what we started, and we also applied the same method of transforming the MRI activity into the DNN feature space, like the output of each different layer. And then we started comparing with two things. One thing is if we pass the same image, the same blurred image to the neural network, how does it compare? So that's what's called RS, and it's represented here on this x-axis. And the input to the neural network was the original image, like the non-blurred image that is coming, like the one that is not blurred. And that's seen here is the RO, so the similarity between the features of the same image that is presented to the subject and the features of the original image, or like the non-blurred image. And the interesting part is that we find that for many different images with different blur levels, that the human brain was actually showing more similarity to the original image and not the same image that is being shown. So what the brain is basically doing is that it's using this feedback information of like, because it knows what a cat looks like. So it starts to project that and to try to make the image sharper in order to be able to recognize it even more. And that's what makes the brain more robust than a neural network, is those feedback connections that are actually feeding back information about what do I know about this object. So all of that is still in the realm of neuroscience. We haven't yet touched onto like the idea of mental health. So, but this is actually where the connection happens. So if you look at those two, like the Pac-Man image and like this vase face, so those are very well-known optical illusions. So even though we have this, like it's just a 3 Pac-Man and like a couple of lines, we imagine as if there's a triangle, a white triangle that is hiding all of like, a complete triangle or 3 circles, etc. And at the same time, also we can, like, it depends on how you start to look at this. Either you see a vase or you see 2 faces looking at each other. And this is actually happening because of this feedback image, or like this feedback drive that is projecting. Is like your brain is basically trying to look for something meaningful in an image that has no meaning. So it starts to generate this whole optical illusion. And there is actually, the idea is that once you have like this optical illusion becomes like in an overdrive, then what happens here is that you start to see hallucinations or psychosis. And there's 2 hypotheses for this idea. Is like you have either your brain is like, if you like, for example, like this hallucination, your brain is either over processing the images that are being shown in front of him. So like, for example, the feedback is lacking, or the feed forward is really strong. So basically you're over processing everything that is around you, that you're seeing things that don't exist. Or the other possibility is that you actually have your feedback system is the one that is too strong, that you are actually projecting your internal thought onto your early visual areas that you're seeing, that is basically, so you're creating something that does not exist. So those are the two hypotheses of something that is like the reason why hallucination happens in something like such as schizophrenia. So at the time when we had this study done, that's when I moved to the Kyoto University Hospital, and we started looking at this idea, same idea, but we wanted to look at how schizophrenia patients compare to healthy controls. And we actually found that it looks like the level of this feedback or this sharpening effect is actually higher in schizophrenia patients. And yeah, so this is like the first thing that we found. So we're looking, actually, this is still something that is, almost getting ready to be submitted for publication. But yeah, the main idea is that we found that the schizophrenia patients, what happens in them is that they actually have this increased level of sharpening in comparison to the healthy controls. Yeah, so this could actually give us an idea of like how is the connections inside of the brain are perturbed, or are having like there's a problem with those connections that are not typical for a healthy human being that is causing those problems. And this can lead us to, like find targets of what we can actually do to either cure those problems or to just alleviate this effect. Okay, so yeah, so the question is, of course, what else? So this is one example that we've been working on, which is the idea of schizophrenia. But schizophrenia is not the only disorder that is accompanied by hallucinations. So we can actually study this with many other different disorders. So such as like in this idea, so like the idea is that visual tasks itself are very versatile, that you can use them to test different brain functions, such as decision making, or like elicit emotion if you're showing like emotional faces, or something like emotional movie clips, etc. So for example, here they're trying to show in this image on the right sensory evidence. So if it tells you the next fish is going to be either from the right or the left ponds, and the size of the bucket in number 2 is telling you the probability beforehand. And then you show the actual ponds, and then you show a red fish, and then it's telling you like, okay, so which pond did this come from? And then you tell them like, okay, I'm confident it's coming from the left one because there's more left fishes in that pond. So this is, you can actually you're not only limited to the visual thing, but you can also go to like decision making, and visual tasks are helping that. Other ideas is also the idea of studying neurodegeneration by modeling using deep neural networks. So if you have actually neurodegeneration or an injury in certain parts of the visual cortex, since the neural networks are really good at doing that, so you can actually try to remove those connections and try to simulate what is going to be happening in the brain. Yeah, so there is like a lot of different possibilities that you can actually explore. And yeah, so that's, I think, all about that subject. Yeah, so of course, in the end, I want to thank, like the Kamitani lab members, which, like I did my PhD and they helped me with all of this, and also Takahashi lab members, where we did all of the schizophrenia work. And yeah, the people who were running the scanners as well in Kyoto University, and of course the funders. Yeah, and in the end, I wanted to give a shout out. So if you want to learn more about computational modeling of vision, then maybe consider applying for Neuromatch Academy and conference. Basically, it's a summer school. It's currently running, so the application is finished for this year, but they do it every year. And they have computational neuroscience and deep learning schools. And also, if you're an Arabic speaker, we have a community for Arabs in neuroscience. We're trying to, increase an underrepresented group of people in the field as well. So yeah, and thank you.
Amazing. Thank you so much for sharing. This is super, super insightful. I personally don't have any background in comp neuro, but learned a lot today. So thank you. Everyone, if we could join in giving a physical or virtual round of applause for Dr. Abdelhack. Yay.
Yeah.
It's always so fun on Zoom. Wonderful. Well, folks, if you have any questions, feel free to raise a hand and unmute yourself or drop them in the chat and I can read them out loud. Give it a little bit of time. I know people take some time to think and type.
Drink some water.
Yeah. Can people connect with you on LinkedIn or social media?
Yeah, so basically, let me put this. So this is basically, if you'll find it on Twitter, on LinkedIn, like everything is basically using this handle, even like the website. It also has like all of the links there. So this is my personal website as well. So you'll find like my Twitter account is there, LinkedIn, everything that is you can connect to, even my email.
Perfect. Another question while we're waiting for folks to chime in. Do you have any advice on students who are looking to travel abroad for studying or looking to seek employment outside of their home country? Any experiences navigating the academic side of it or even the cultural side of moving to a new place?
Yeah, so it depends on where you want to go, because the way that you navigate the system is very different. So for example, North America, USA, Canada, the whole thing is that you want to find someone who you're interested in working with them, like basically a PI or principal investigator. So you're going to be, if you're going for grad school, of course, if you want to work under their supervision to get your graduates, like mainly PhD, because that's where you're going to be finding funding. Masters usually are not funded and it's quite expensive. But yeah, so the idea is to find those people and then to connect with them. So most of them have lab websites, or even you find them on Twitter. They are advertising their positions, especially Twitter, not LinkedIn. I mean, some do LinkedIn, but mostly Twitter. Yeah, so those people, like, maybe try to hit them, or maybe you wait for them to announce, hey, I'm looking to hire PhD students. That would be like the perfect timing, of course, but sometimes they just don't, like, if they find someone who's really good, they will just try to get them. So yeah, the important thing is when you're contacting them to actually read their work before you contact them, because, just a generic email will not cut it. They receive like hundreds of those emails every day. So you need to have something that really attracts them. And at the same time, the challenge is how to actually put enough details that shows that you actually understand what you're talking about, while keeping the email short, because they also receive like thousands of emails per day. Yeah, so that's the point. And of course, you attach your CV with that as well. Japan, for example, where I studied, mostly it goes with what's known as the Max Scholarship. That's like most of the international students in Japan go there through this scholarship. And for that, they usually open the applications around May, April, and it's usually in the, the Embassy of Japan in your country. So yeah, hit them up and they do all of these things like in there. And in that, you don't even have to contact the professors beforehand. They will just you get the scholarship, and then in the part of the process, you actually start to contact the professors, but you don't have to do that in the beginning. So they will do like some math exams, etc., so that to see whether you can you qualify or not. And that is both for undergrad and postgrad for most of the countries. Europe is, yeah, Europe usually they have, like, so it's very similar to the US. You also want to contact the PI first, but they also have usually those things are announced as well, because they have like a legal obligation to announce those things, if I remember correctly. And yeah, in Neuromatch, we actually last year we held a session about how to apply for grad school, and there were different professors from different parts of the world that were talking about that. So yeah, if you'll have to dig it up though. I mean, I will probably also have to find it out where it is. But yeah, if you're interested, maybe yeah, just hit me a message or something, and then I'll send you the link.
For sure. Thank you for sharing.
And maybe they will hold another one this year because it was really popular.
Absolutely hope so. Fingers crossed. Very, very short plug for the Neurothon workshop series happening Monday through Friday this week. Some of this workshop hosts coming in are also international students. So could be also great to ask for their advice, since I think they've very recently been through the, one of them is based in India, applied for admission in the UK. So maybe has like very, very latest cycle information as well.
Mhm.
Stay tuned for that, folks. I hope I might be mispronouncing your names. Please correct me. So, okay, has a question about any books you recommend for neuroscience.
Yeah, so there's this book called Fundamentals of Neuroscience. So let me see if I can get if I just want to make sure that I'm getting the name correct. Yes. Fundamentals of Neuroscience. No, it's not this one. Oh, fundamental neuroscience, not fundamentals of neuroscience. Let me put that in. If you want like an easy read, or like that is going to like the idea of computational neuroscience also. So there is this really nice book there, like by Grace Lindsay. It's called Models of the Mind. It's a really nice book. So yeah, and Grace is a brilliant person also. She's starting to become like, she's going to be an assistant professor really soon. So yeah, it's like she does know her thing.
For sure. That's great.
She was my mentor at some point.
Oh, no way. That's amazing. Are there any more, I don't want to say popular science, more of kind of, a lot of folks have like Goodreads lists of neuroscience books to read or anything. Are there any books that are like very casual reads more so have caught your eye in the past?
Yeah, this Models of Models of the Mind. That's the one I would
Okay.
recommend.
Yeah. Awesome. Okay, it took a while to load that link.
I think there's someone who's writing a question above my link.
About the link?
Yeah.
I think Matthew has a question about some of the research you were discussing as well. I believe maybe the science book has quite a hefty, we love the hefty textbook prices, but sometimes there are online repositories that are easy to access as well. So online libraries at borrowing.
Yeah, and maybe if you hit Grace herself, like she's on Twitter also, maybe if you try to ask her, maybe she would give you a copy.
Absolutely.
The difference between lack between computer and brain processing, clarity of the images. Oh, so you're asking about why don't the images look exactly like the brain? Is that your question?
Yes.
Okay. Yeah. So first thing is fMRI is really noisy. So you're scanning, plus it's also the resolution that you can scan by. Basically, as I said, it takes like maybe 2 seconds to scan like the whole brain. Of course, it depends on different types of scanners. But this is given a resolution of like 2mm by 2mm. Also depends on the scanner. You can change those. But the thing is, like, if you do, so that's basically a couple of hundreds of neurons in that space, or maybe even more. So if you decrease that, then you have a problem that you might be increasing the noise. So that's like you're having, and also if you shorten the period of time, you're also playing with this signal to noise ratio in order to get a meaningful signal versus just getting complete garbage. And yeah, so you have a problem with that noise, and you also have a problem with the spatial resolution. So you're not getting like all of like one neuron or the output of each neuron separately. So that's the answer to that. Number two, you mentioned our brain has a kind of feedback mechanism to fill in. Is there a way to introduce this process? Yes, there are people that are currently working on that. We actually tested a toy model in after this, and actually this toy model was really interesting because it did not really improve the way that the model saw the sharp images, but it improved really a lot as you introduce blurred images, even though the model was not trained on any blurred images. So yeah, and there's a couple of studies that were looking into that. Yeah, I'll have to, if you want, I'll have to find in the library. So yeah, if you want to get some of those links, yeah, also hit me up. I'll find them.
Thank you. Any other questions, folks? I don't see any hands raised. If not, like Dr. Abdelhack said, please feel free to shoot him an email. He's happy to connect you with resources. And just want to say thank you once again for joining us. I know it's been a very busy weekend.
Thank you.
So really appreciate you joining us this evening. Yeah, everyone, hope you had a really great time learning more. Comp neuro, deep vision, wonderful, wonderful list of topics.
Thank you. Thank you everyone for coming, and sorry to keep you up all this late. And yeah, have a great week ahead of you.
Thank you. Take care.